My name is Japie from Westhuizen. I'm General Manager of SA Studbook here in South Africa, and I would like to welcome you at this beef cattle breeding course. It's the first section. It's also divided up in a few parts to make the video shorter. This is basically a combination of a course that's been developed over many years with the assistance of course goers and farmers and breeders and also students. My co-author on this course is Dr. Helena Tron, also working for SA Studbook, an animal geneticist in our service. So we can kick off then with telling you more about some basic genetic principles. The content will start off with what we would know as genes, what heritabilities are, pedigrees of animals and how that actually fit into breeding, the value of contemporary groups, and then very important also traits, breeding values, and eventually also selection values. I would therefore like to kick off these lectures with just sort of giving a brief overview of what genetics actually does and how it will change the phenotype or the visual evaluation of animals and what we see in terms of genetics as well. So this is just a slide to illustrate the vast difference in one species, cattle, and that we can expect just simply due to genetic differences amongst those cattle and what we expect. On the top left-hand corner, we see an example of the Belgian blue. Obviously, one of the breeds where double muscling or the double muscling gene is well known. Here's the example of the Belgian blue. Then an African breed, the Nkone or the Watusi cattle. The Scottish Highland, you can see very well adapted to cold and windy and wet conditions. The Gur, which is a Indian breed, a milk breed, was Indica's breed or a Zebu breed. The Hereford, that's very numerous all over the world, known for good quality beef production. Holstein, top dairy animal, milk production. The Shorthorn, that's given rise to a lot of other breeds. Some of the composite breeds has been developed out of the Shorthorn, and the Angus as an example of a prime beef producing animal. And then at the bottom right, you can't see it very well because of my picture being in the way. Also a Beefmaster or a Boeran, maybe in this case, beef producing animal. Okay, let's kick off with a few principles. The first thing is the mode of inheritance of all live animals, obviously, that you find, and even plants and others as well. And we know that the code for the genetic differences are carried inside each nucleus of each cell that's part of the body. This is basically on long strings, chromatide strings with a specific code. We call it base pairs, and these codes can be either A, C, G, or T that depicts the genetic code within these chromatides, and the chromatides are then wound up into so-called chromosomes. That comes in pairs. We'll talk a little bit about that later as well. Pairs of chromosomes in each cell, except for the sex cells, the semen and ova. It's very tightly packed DNA, and a long string of this code then we would call a gene that carries the genetic code of how that body would behave under certain circumstances. It will moderate the effect of, let's say, heat and growth rate. It will also contribute to the phenotype or the measurements or even the visual assessment of animals. And this code is basically causing all the variations in life. This is an example of the chromosomes then. This is for cattle. So we have, they come in pairs, so chromosome 1, that's got the chromatide strings on it with the genetic code. They come in pairs, obviously Y in pairs. You get half your chromosomes from your sire, or the animal from its sire, and half from its dam. And that's so in the sex cells you just have half the number of chromosomes. They don't come in pairs. And you will see in the case of bovine or cattle, you will have 29 chromosomes, non-sex chromosomes, and then one pair, the sex chromosomes, that could either be an X or a Y, as we call it. And you will see in the case of a male, you will have a X and a Y chromosome, and in the case of females, you will have two X chromosomes of the same size. Like I said, the Y2N, we call it 2N, or that they come in pairs. You get half from the sire and half from the dam. So at where they basically then, when mating takes place, you get ordinary cells again, body cells that's got the pairs linked together. So when we estimate the breeding value or predict the genetic merit of an animal, it's therefore very important that the parentage should be known. So the pedigree information is basically being used because an animal can only get its genes from one source, and that is from its parents. So each of the parents will contribute half of the chromosomes, and therefore the genes with the genetic code on it that the animal has. Chromosomes would also be a cause for variation, in the sense that while the sperm and eggshells are being formed, and these original cells split into two similar cells. Remember, originally they will have a pair of each chromosome, and carrying the genetic code, and in a sex cell, that's a sperm or an oocyte, an eggshell, there will only be half, so those cells need to be dividing in half. And whilst that happens, chromosomes small pieces of a chromosome can tear off and stick to the other side. So like I said, they come in pairs, so your opposite pair would then actually get a piece of the string of the chromosome, and that causes variation. So nature is actually focused on variation, so that progeny will always be adapted to maybe changes in temperatures or changes in the environment, and that variation means that there will be animals amongst them that will be adapted to these changes. A lot of the tests based nowadays on whether animals are carriers of specific genes or forms of a specific gene, and also to maybe verify parentage, is based on the fact that you can read parts of the code on these chromatides or these strings with specific codes on them. So this is being used, for example, in with so-called DNA testing for parentage, and this is just an example where you have two possible sires, sire 1 and sire 2, and this is the dam. And you will see that there's a specific calf in this case that was born from the dam, so you can see it actually resembles parts of the genetic code of the dam. And if you then have to distinguish which one of these two sires are the most possible probable sire, you will see that sire 2 hasn't got the same parts of code that the calf has. And that means that it can only be sire 1 in this specific case. So this is being used on a daily basis by lots of breeders all over the world to verify parentage, especially the sire of calves. Just a few words on so-called genomics. This is now an example of the string. So this is a long string of a chromosome or the chromatide strings, and in normal cell division, it will actually rips open like a zip from the one side, and there are pieces floating around inside the cell that will actually stick to it, and these so-called letters T, G, C, and A will always bind in a specific manner. So A will always bind with T, and C will always bind with G. I mean T and A, and C and G. And these strings can also then form proteins because of amino acids being part of this. So in a normal cell division, this will also happen to form new cells and to form new strings of these chromosomes. So in this process, it can sometimes happen that these codes can change. And with changes in codes, it means that there's also a new variation being formed if these changes result in proteins not being able to form, or there's a mishap in these coding that changes. It could lead to deformities or sicknesses, congenital deviations, and so forth. But in the normal sense, it doesn't happen. There are certain changes that can happen, that the code would not change the type of protein, but that would then distinguish one animal from the next, because the code would not be the same, even if they are from the same breed, the same family, and so forth. So we call these changes so-called mutations. So a mutation will then cause a difference in the DNA code. So no two animals in the world would be exactly the same, except where you have cloning. And that means that you can actually identify an animal. It's actually beyond a fingerprint. It's more accurate than a fingerprint to distinguish two animals from each other. So in certain cases, there could be a wrong base with the so-called messenger RNA. This we call DNA, the normal strings. RNA, these messengers that takes the code out of or read the code from a point and then form amino acids, and these amino acids then wind up to be proteins. Our hormones, for example, in our body are proteins, and these proteins then govern basically the response of the body on different things. How much feed does an animal need to eat before it can gain 1 kilogram? When will it be comfortable in terms of temperature? And so forth, and so forth. Fertility differences amongst the animals, the color of the hair color, of the skin color, the pigment in the skin, and so forth. Those are all part of the code of that. So one word that's often being used is SNPs, and a SNP is basically a place where there's variation on the code within the same species and breed, due to the possibility of mutations that took place earlier somewhere in the breed, and so the genomic information that we currently use is the labs would actually, or the geneticists would actually look for the place where these so-called SNPs, so these are places or locations or loci on the chromosome where these SNPs or these codes would differ amongst the animals. So there's a T, A, G, and C, and that's then being used because of the variation. There's a lot of code that's exactly the same. You will see it's exactly the same. So it's only on where these mutations took place somewhere that you can actually use that to your advantage. How do we go about? This is just a practical thing. A farmer would pluck usually tail end hair, send that in. That's the DNA is being extracted, or the genetic code is being extracted. There's a very interesting chemical process on these little panels. So this is a typically genomic panel or SNP panel. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So 32 animals DNA can be done on one of these plates. The DNA is basically shredded up in small parts, and there's pieces of DNA sticking off little beads that's on these little platelets here, and then they attach to these little beadlets. So on a specific plate, you will have in the region of 50,000 or more of these little holes with little beads in it, and then they illuminate in a certain color. And then you basically have a computer reading of this little bead, or all these beads, the colors, and that would tell you whether you have A, C, G, or T, or a specific form, 0, 1, or 2, on a specific location on the chromatide string. There's very interesting applications. One of the applications is that once you know each animal's code, or each genotype as we call it, you can also distinguish the distance, the genetic distance amongst these animals, how related they are, genetically related they are to each other. And these genetic distances or relatedness can then be plotted on a three-dimensional graph like this example. So this is a scatter plot, just giving you an example of how distinct or different these breeds on the plot are from each other. So there's just the Holstein, the Bonsmara, Drakensberger. It's a 3D plot, so you can actually, if you work with it, you can actually rotate it so that you have an X, Y, and Z axis on this. Obviously, this is also being used in terms of parentage. Of course, you can determine the actual genetic relatedness amongst any group of animals. This brings us at the end of part 1 of this presentation. Good luck, and we will then later on continue, or you're welcome to look up part 2 of section 1.